So I think we are ready to go again. I see that everything seems to be working. Hi, hello, and welcome everyone again. Microbehunter here, Oliver here. And welcome to another Saturday live stream. Sound is good. Today I would like to talk, as requested last week, I would like to talk about oil immersion microscopy. Not the first time that I'm talking about this topic, but there are always a few new variations to the topic. And additionally, I would like to also show you how to do so-called heat fixing of slides, because sometimes this is also necessary if you want to do oil immersion microscopy. I'm just going to show it to you today. Well, as always, I would like to send a hello all around the world. I would like to invite you to, of course, ask questions in the chat. However, I will not be answering them right away because I will first simply do my program as I kind of planned it, a little bit improvised sometimes as well. Feel free to ask questions, and then after 1 hour approximately, I'm going to try to answer the questions in bulk. The reason being so that it kind of does not interrupt the flow of the live stream too much. Okay, for the last 2 live streams, that's the system that we tried out. It seemed to have worked. I will try it to keep it like that. And another thing that I would like to simply mention is the following is, if you want to, obviously, I've now also, it's possible to also now donate a little bit of money. And if you do that, then I will use this money to buy some antique microscope slides, which I will also then present in the live stream. So there's this called the stickers or the super chats. So yeah, I would like to simply also mention that. And also another advertisement a little bit here is I also have a microscopy shop, Amazon affiliate shop, if you would like to visit that as well. Have a look. I'm going to try to keep the products updated as well. So I'm going to jump directly into the program a little bit, what I wanted to show you. And I would like to usually use the first couple of minutes to talk a little bit about the theory, and then I'll show you some practical stuff as well. What I've got here is, look at this here. Immersion oil, okay, and of different types and brands and so on, and not all of them work. Why not? Because look at those here, those immersion oils, and this is already the first point that I kind of want to make before I actually tell you what it's good for. Look how do you like this bottle of immersion oil? Do you see this yellow liquid in here? Look, it has completely turned solid, and totally not usable anymore. It's like honey. So this is actually one of the problems with oil immersion microscopy, that some immersion oils, the so-called the natural immersion oils, sometimes tend to solidify over time. Okay, so that's something I'm going to talk a little bit about. And over here I've got 2 bottles of synthetic immersion oil, and they have remained liquid now. This one, for example, since the year 1998. So for a very long time, it's still usable. Okay, so I'll be talking about this here as well, but simply to give you a little bit of an insight of what I have here. And I would like to do the following is I would like to start out with a little bit of theory. You got to bear with me, okay? But I did prepare a little diagram so that you understand what the whole topic today is about and what oil immersion microscopy is. So for a couple of minutes, I have to explain this to you, okay? Let me rearrange the camera a little bit. What I have over here is a schematic diagram. This here is the objective. It's the oil immersion objective, like the one over here. Wow, thank you very much. I see that people already start donating. Thank you very much. And again, I'm going to use this money, and I'm going to buy some slides that I would like to show you. Yeah. So that's the oil immersion objective. Over here, you know it's an oil immersion objective because there's oil written on here. And here I show you 2 cases. Okay. So the left case is without immersion oil, and this one is the cover glass. So there is usually water in here. Right, the orange dot here, that is the specimen, and this is the microscope slide. And when the light goes, and these are the lines here are the light rays, and if there is no immersion oil, then you can see that because of the difference in refractive index of the glass, the cover glass, and the air, the light rays that are emitted by the specimen kind of are refracted and bent outwards, and in this case they're missing the objective. Yeah? And this basically means that the image is going to be darker and also of lower resolution. And if you add now immersion oil here directly on the objective, and you see that the objective is actually covered in oil. Yeah? So this light blue part is oil. Then the light will travels in a straight line, more or less in a straight line, and it is still able to go into the objective. So the resolution is better and also the brightness is higher. Okay, so this is the reason why you use immersion oil is to make sure that the light does not have to go from glass into air, because this will actually cause a loss in image information, right? And this basically means that what you have to do is you actually have to take a drop of this immersion oil here, put it directly on a microscope cover glass, and what you're doing is you're actually rotating the objective into the oil, so the front part of the objective will be covered in oil. Yeah, and so that is when I first did that many years ago. I could not believe this. What are you actually want to tell me that the objective is going to be actually covered in oil? Yeah, and this is that's indeed the case. And here I would like to already show you, and maybe if you just compare this with a non-oil immersion objective, just so that you actually see that this one is a 100 times. Yeah, this one is a 10 times. It doesn't matter. But if you just look carefully, what you're actually going to see here that there is over here this white thing here it's sealed off with some I don't know, it's silicone or something or paint. It's completely sealed off. So this basically means that there is no possibility, if you use the objective carefully, for the oil actually to enter the optics, go in here. And look over here, if you just use a regular non-oil objective, then you see that there's of course a little gap in here. Yeah, and this basically means, and also the front lens is not oil tight, so that means oil can enter. So the important thing is, and that's one of the reasons why the first warning, only use immersion oil on oil immersion objectives. And you know you have an oil immersion objective because oil is written on it. It's so important. Why am I saying this? Because I received emails, and of beginners who basically were suggesting, well, I have a problem seeing the specimen, so should I now use immersion oil also on the other objectives? Seriously, if you do that, you end up actually destroying your objective. Okay, so you got to be really careful about this, that you're not using oil on non-oil objectives, and this can happen very easily. Also happened to me actually. If you kind of use oil immersion, right, and you're observing the specimen, and then you change the magnification back to a lower one, to let's say 60 times or 40 times. Because you're not thinking, or you just forgot about this, and then you're rotating the 40 times or 60 times objectives also into the immersion oil. This can happen very quickly, also happened to me. So when you're using oil immersion, and once you've got the oil on the objective, do not change magnification anymore. Okay, otherwise you end up actually messing up your objective. Okay, funny story? No, actually not so funny story, but an anecdote. A couple of years ago, I talked to my microscope representative who was selling me the microscope, and we were because I'm a teacher in school, and we were talking a little bit about microscopes for use in schools and education. And I said, this is actually we need microscopes, but without oil immersion objectives, because students you shouldn't be using those, right? Because they end up messing up the other objectives as well. And that's why I told the guy that we remove all of the oil immersion objectives from the microscopes, and we also remove the immersion oil, so that no student can get the idea of actually using those. And then he started to laugh, and he said, doesn't only happen in schools, but we're currently fixing an objective for a research institute. And this objective was several thousand euros worth. Those highly specialized objectives are really expensive. And actually, it happened that immersion oil went into the non-oil objective somehow behind the lens. It was so messed up that actually the oil went into it, so that into the actual system. So they had to disassemble the whole objective and get it cleaned. Yeah. So he said this doesn't only happen to students, and can also happen to quite experienced people. So I'm just saying, that's why I'm not a big fan of oil immersion, because one has to be careful. Okay, so this is a little bit a thing that I would like to say. Yeah, if you've just joined the live stream, Like in the previous times, I would like to invite you to ask questions, but I will only answer those questions after approximately 1 hour. Okay, so this is already another thing that I need to say. Look at this here. The immersion oil will essentially, the natural immersion oils, and you know that it's kind of natural because of this yellowish color, will over time start to solidify. And this basically means that you have to properly clean the objective after every use, okay? Because otherwise with time it's going to collect dust and dirt, and it might also turn solid. The situation is a little bit different if you use synthetic immersion oils, like the one that I have over here, because they kind of remain liquid. Okay, so there is a difference. There's a difference, yeah. This is basically something that I just want to inform you about. I also want to show you something else that I kind of consider a little bit remarkable. This is when you buy those microscopes with oil immersion objectives, they frequently come with those bottles here, and you don't really know what's in it. Okay, I don't know which refractive index it has. I don't know what substance it is. If you buy oil from a known company, okay, look what you're going to get along with the bottle. I just want to show this to you, okay? You're getting a full manual with just for the oil. Yeah, it's a safety data sheet. Yeah, it's 1 page, just or maybe you can see it here, 1 page, 2 pages. Yeah, 3 pages, 4, 5, 6, 7 pages. Yeah, okay. Yeah. So that's a safety data sheet, yeah, with all of the technical details, yeah, of the oil, yeah, the density, the boiling point, everything is properly documented here, right? So that's actually something nice, yeah. And then sometimes when you buy stuff, you don't even know what you're getting. So I'm just should be kind of just saying. What I would like to show you now is before I start doing a little bit of the practical stuff, I would like to show you a video that I made of a really bad. Oh, thank you again. Okay, thank you again for the support. Okay, I just want to show you a very quick video here that I've made of a very badly solidified immersion oil. Just look at this here, okay? This was actually, pulled this out from my school, somewhere in the very back somewhere. I found, totally new bottles of immersion oil, never used before, and for maybe many years. Look how solid they have become, yeah? So this is, I unscrewed it, of course, yeah? And just to check, and I've seen actually the bottle was never opened before, so it was still sealed off. Yeah, there's a plastic tip that you have to cut off. Yeah, but still it turned solid. We didn't throw it away because it was kind of kept it as a souvenir. And I also tried to open the bottle. Yeah, here and look how solid it is, because some of it is actually solidified right here in the lid as well. Yeah, it's like glue, kind of. Yeah. So that's basically why this should be a little bit of an indicator or a warning that you have to properly clean your microscope objectives afterwards. Okay. Just wanted to share this with you. Some kind of extreme cases. Yeah. So here we're starting again. So what I would like to do now is the following. I also would like to show you now another thing that I'll be using on my microscope, and that's the so-called focus lock lever. And where do I see that? That's the one over here. Okay, here. Because not all microscopes have that, but it's a very convenient thing to actually have. And that is next to the coarse focus knob, there is this lever over here. Okay, and basically what you do is when you set this lever up here, okay, and when you're lowering the stage for defocusing, and when you're rotating it again, then it will basically not move higher than the setting over here. So what you can do is you can essentially, when everything is focused, you can lower it, you can add the immersion oil, okay, to it, and then you simply rotate it back here, and yeah, you'll be fine. Yeah, so this is a very convenient. It's a very convenient tool here, and but when you move it back, then you kind of unlocking it, and then you can actually move it yet further. Okay, so this is a so-called the focus lock lever that allows the microscope kind of to remember the focus position, which is very practical if you're using oil immersion microscopy. Yeah, so so this is something that I'll be using in case you're wondering. But in the meantime, what I'll be doing, and sometimes the camera goes out. Yeah, see, sometimes the camera has gives me some problems here, and I will try to reset the camera, hoping that this works. Yeah, here we go again. There seems to be some kind of a slight connection problem. Okay, yeah. I can also refocus the camera here again. So I've done this so that you see better what I'm doing here, right? So this basically means that I'll be using, I'll lower the here, I'll be using this objective over here, and you see that over here oil is written. Okay? So this is the only one where I'm allowed to use the oil immersion. Yeah, but of course I'll be starting again at a low magnification. Yeah. So next thing that I'll be doing now is I'm going to now make some simple slides, and later on I'm going to show you some of the heat fixing as well. So when making slides for oil immersion, first of all, also just as a warning for those of you who are maybe new into the topic. For the others, you just have to bear with me again. Okay, you are not. Don't ever think. Don't use oil immersion on permanent slides. Okay, sometimes I'm saying the obvious, and there are several reasons for this. First of all, they're extremely difficult to clean. Okay, so if you've got permanent slides and it's covered in oil, how are you going to get the oil off? So you have to kind of rub it with some tissue paper, and you're exerting pressure on the specimen. Not good, right? So that's difficult to clean. Number two, the oil that's on here, if it touches the slide label, it's going to mess up the slide label as well. Okay, also not a good idea. And reason number three, that some permanent slides are actually quite thick. Okay, however, that's also not good. Because if it's too thick, then because the distance between the objective and the cover glass is so small, if it's too thick, then you're going to crash into the cover glass. Thank you again. Okay, trick track. Thank you. Okay, so this is something where I'm saying don't even use it on permanent slides. Yeah, so. If you want to make a slide here, I've prepared a couple of samples. I've got some chlorella algae. I've got bacteria. Now you're gonna say, oh, where did I get the bacteria from? I can show you if I can find it. Where did I get the bacteria from? Where's the bottle? Where's my bacteria? Here it is. Okay. Again, I bought this here. I only want to work with safe bacteria, right? So I bought myself some time ago this bottle of bacteria here for making yogurt. Okay, thank you again, Pat. This is for making yogurt, and this basically contains bacteria which are not dangerous, right? The bacteria are freeze-dried. This basically means it's a bacterial culture which was frozen and then vacuum was applied to take away the water. So that's a way of preserving bacteria. And most or many of them don't die in this process. Some of them might, but there's still a lot that survive, right? So these are freeze-dried. So it's basically, maybe some of you have also heard of astronaut food that astronauts sometimes eat. It's also freeze-dried. It's basically completely dehydrated, free of water. Okay, and this basically means that those bacteria are basically safe to eat. And what I've done is I've taken a little sample here a few hours ago, and I've essentially added it to a little bit of water and allowed it to suspend a little bit, so that the clusters kind of break apart. Yeah. So, what I'm going to be doing is I'm just going to go through this here, and I'm going to start making a slide here, and I'm also going to give you a couple of tips here as well. Okay. So, very important. Yeah, you can use a pipette. Very important is do not use too much liquid. So that is really okay. I hope I'm getting now some of the here. And when I say not too much, I'm saying the cover glass should really stick to the slide by by capillary action, so to say, by the adhesive forces. So no floating of the cover glass. First of all, because it's too thick, then the cells are going to float up and down vertically, and you're going to lose focus, yeah, especially with oil immersion. Number two, if it's kind of floating around and there's oil on top, then the objective is going to kind of move the cover glass around as well. Yeah? So very little liquid, then a cover glass. Okay, then a cover glass, as always. And what I usually do is when you look at it, difficult to see a little bit is it doesn't even fully spread beneath the whole cover glass. Okay, and you see that it's pretty stable. So, and we're just going to do some regular microscopy using this here, and yeah. You can actually see the thing, so that's already the 10 times. Normally you always start focusing with the 4 times. And I have to open this here. You still see a whole bunch of clusters. The chlorella algae were also in tablets. And the thing that kind of surprises me a little bit, and I don't understand this, there is even some movement going on here. You see this? And I do not understand this, because I'm going to show you now the following, just a second. Because these were the ones I used. The chlorella algae are tablets, right? And basically I can also show you, I mean, these are just green tablets, and I just have taken one of those here. Yeah, tablets, and I scratched a little bit off, I dissolved it in water. And I don't know, are they still alive? I don't know, this is kind of interesting that they were able to survive this whole process, because they're moving around. I don't get it. Yeah. So, yeah, you see there are a whole bunch of air bubbles as well. Okay. I will put in the, yeah, I'll just go up with the magnification. We're just going to find maybe a place where they're still clustered together. Okay. And maybe the arrow, I just move up. It's now 40 times. I go up with the... This is not normal. This is... Look at this. No. There must have been... This must be a contaminant. I'm totally baffled, because this is a rotifer. It's not supposed to be in there. I mean, I used water from this water bottle over here. So maybe I got contaminated. It doesn't matter for the sake of our demonstrations. It doesn't matter. But this is, I must have gotten some contaminants in here, because this is not okay. This is somewhat unexpected. Okay, but again, this is a little bit improvised. Okay, so 60 times. Look at this. I must have messed up something. Maybe I did not wash the petri dish properly. Look, do you actually see this here? There's this the scale of a butterfly over here. Okay, my bad. I think I must have. I don't know. Yeah, doesn't matter. We still have. This basically means that we have something, a few more interesting things to observe here. Okay. Now look at this. These are not. I don't know. Okay, I'm a little bit baffled right now. Okay, so check up here. I've got my 60 times, and now we're going to do the following. Everything is in bright field, so I'm going to leave it at bright field, because many of you know that I like to also do a lot of stuff using differential interference contrast, like this one over here. So a little bit of color, right? Looks beautiful, but most of you probably, yeah, just would like to see how the whole thing looks in bright field, so that you can actually compare it with what you have yourself, right? So, and now I'm going to do the following. I'm going to put the oil immersion objective into place, and this is how it looks like. Okay, it's a little bit out of focus. Let's go put it in focus. This is now oil immersion objective without oil. Yeah, it's kind of blurry. Yeah, and we can also remember the brightness a little bit. Okay, I'm going to put this away. Okay, and now I would like to add a little bit of immersion oil. Okay, what I do now is I'm going to basically, now we have to be a little bit careful. Maybe I'm going to show you this over here so that you see it a little bit better. You see a little bit in small what we've got, yeah, in the microscope. I'm going to now move it over like this. It's kind of very bright. And I'm going to now use the immersion oil. And I'm going to apply a drop of this immersion oil directly on the cover glass. Okay, you might not be able to see it because maybe the bright, yeah, there is a drop of immersion oil now on here. Okay, I do the following. I now rotate the objective into the oil. It's covered in oil. Okay, I turn up the brightness again a little bit. Yeah, I have to then refocus. I'm going to go back to the scope view. Okay, and I think you're going to already see that it's significantly more crisp. Okay, so we're now looking, yeah, at the whole thing with the maximum magnification. I can, of course, still move around. Okay, this kind of already shows why it's important to keep the amount of water very low, because if you look at some of those cells that are floating around, they still are able to move in and out of focus as when they move vertically, because the depth of field is so small. Yeah? So this is, yeah, just for trying it. Let's now try to look at the whole thing in DIC. I have to change the filter around. It's a little bit dark because I'm already at yeah. And this is how it looks in DIC. So, but, and this is again a regular bright field. Yeah. So this is basically now the time when I'm now going to engage the focus lock lever, which I just showed you before. Okay, everything is in focus. I pull the lever forward, which you cannot see. Okay, and now what I can do is the following. I can now lower, just to I'm going to demonstrate this to you now. I can now lower the stage with my coarse focus, and when I go up all the way and let go, then I'm again back in focus. Okay, and now I lost the oil film a little bit. Okay, here we go again. Yeah? But that's kind of the convenient aspect, convenience. It's kind of remembered now the position. I go out, I could coarse focus. I could exchange now the slide, and I basically move back into focus again. And there is no risk or no danger for actually crashing into. Okay, this was the oil film that started to form again. Yeah, and there's no danger for me of crashing the objective into the cover glass. Yeah? So this is a very convenient feature. So just for the fun of it, I'm going to now do another slide. Okay. So this was basically these algae. Let's have now a look at some of the bacteria here. Okay. So I'm going to. I can do this now a little bit faster, I guess. Okay, so this is what the chlorella algae, the bacteria here, which are just resuspended in water. I'm going to change the tip. This is a little bit strange, really, that there are so many other cells here. Okay. Yeah, I will take a small sample here again. Okay, we put it on the slide. I drop the tip. Going to wipe this. So here we go. So, and I'm going to try now to do the following. You know what? Why not try to put the oil directly on here? Okay. I'm going to now go to the stage view again here, so that and I'm going to now exchange it. With the coarse focus, I'm going to move all the way down. I take this old slide out, put the new slide in. I make sure that everything is centered properly. I use the coarse focus again until it makes contact. Okay, and let's have a look at the scope view. And Ah, okay, these are not the bacteria, these are the yeast cells that are prepared. Okay, I prepared some, so I switched up the two petri dishes. I added, yeah, these are basically yeast, dry yeast that I added, and basically, yeah, these are the yeast cells. So these are not the bacteria like I thought, but yeast cells that I kind of prepared. And sometimes also quite nice to see is if you add a little bit sugar, and if you wait a few hours, then you actually might see some of them divide. Yeah, they're moving around now. You can see them vibrate. This is all due to Brownian motion. Let's just for the fun of it, let's engage DIC again. I have to, it's kind of dark again, and this is how it looks in DIC in oil immersion. Again, I'm not. If I now were to go change magnification, then basically you don't want to do that, right? Because you're going to kind of dip the other non-oil objectives into the immersion oil. No, not so cool. And then you have to kind of, no, oh gosh, come on. You see this here? What is this? Didn't clean the slides properly. This is starch grain from a potato starch. Yeah. So, because the slides that I'm using right now, I washed them. I did not. They're not new, but I've rinsed them and probably not well enough. Huh? Yeah. Okay. Yeah. So this is basically, again a short demonstration here. Yeah. How the yeast cells look like. In bright field. And you know what? Last but not least, let's try the, I forgot which one I just took. Gee, maybe these here, I don't know. Let's have a look at some of those bacteria as well. I should have labeled, always label your stuff when so I think that these might have been the bacteria. The yogurt bacteria which I bought in freeze-dried. Oh, you didn't see that. Okay, here it is. And let's give it a try. Again here. And again, you see, there is sometimes a little bit of water here. Well, on top. You didn't see it again. Here I added the oil. I have to readjust the camera a little bit. So I go back to the stage view, and I'm going to lower everything again using the coarse focus. I take the old slide out, I put the new slide in, and I use the coarse focus again to raise it. Okay, and indeed, this seems to be the case here. Here we go. So these are now yogurt bacteria, and you can see over here, these are still some clusters of the freeze-dried yogurt bacteria that did not dissolve yet. But you can see that over here we can already see little cocci, or the round cells, and so on. Okay, so this is basically here we go. These are basically yogurt bacteria of I think they put 3 different species of yogurt bacteria in there, and depending on which type of yogurt you want to make, you can buy different freeze-dried yogurt bacteria. And over here again on the side, these are still clusters that did not dissolve yet. But you can see that occasionally those bacteria are starting to break free, and then they're moving around not on their own in this case, but because of this capillary action, and also because the oil is exerting a little bit of pressure on the cover glass as well. Sometimes, look, I'm now focusing back and forth. Okay, and you see that this does not only change the focus, but it also pushes the water a little bit. That's why also the bacteria are moving back and forth. That's because of the slight pressure that the objective exerts on the cover glass over the oil. So just for the fun of it, let's try to see the whole thing again in DIC. This is basically how it looks in DIC. So the thing is now the following, and this is where I would like to show you now a little bit of heat fixing, is sometimes you want to stain the specimens, right? And so what we've done, because at university when we did observation of bacteria, and sometimes there were staining procedures, and we sometimes had to, for example, when doing gram staining, we had to exchange the different stains. So there were multiple steps involved, right? Yeah. And the question is now is how do you do that? Because what we need is we need to have the bacteria fixed on the slide. And I'm going to now show you how to do that. Okay. Again, this is a technique I just want to show you for educational purposes, right? But it's probably not something that you're going to use very often. And I'm just going to use this over here. And what I've done here is I've, for a few hours ago, I put some of the yeast suspension on the slide and allowed it to air dry. I did not even heat it up, I simply allowed it to air dry. We can now look at this here, with another objective. So what I'm going to do now is I'm going to, there is no oil on here, so that's not a problem. So what I'm going to do is I'm going to lower this here again. Well, actually, I'm going to lower this here again. I'm going to remove this slide here. Okay. I'm going to change the magnification and let's raise it again. Maybe we're able to. Let's see if I'm able to find it. See, and that's always you should always start with the, you know what, I'm just going to start with a lower magnification here. Here we go. Okay, here we go. So, no oil on here. These are the yeast cells from before. Okay, but in this case they're dry. Okay. So you can see that they kind of are, because of the drying process, they kind of clustered together. Yeah, but they're now in one plane level on the slide. And what I'm going to do now is I'm going to remove this here again, and I'm going to heat fix them now. And this basically means is I'm going to now normally use a Bunsen burner, and you are going to apply a flame from the bottom side. What happens is that the cells will then start to stick. Of course, they're killed off, but they will kind of stick to the glass slide as well. Why do they stick? Well, it's a little bit like the proteins that are in the cells, just like in an egg. When you put an egg into a frying pan, then it's also going to stick to the frying pan because the proteins in the egg will form cross linkages with the glass, yeah, or the frying pan in this case. And by applying heat, they will also be baked to the glass slide. You do not want to exert too much heat, otherwise it's going to burn, right? And you don't want that either. And when they're baked to the glass slide, then you're able to apply stains on the surface without actually washing the cells away. Because if I were to add now a stain like methylene blue or any other stain to the glass slide, what's going to happen is that they're going to be, the cells are going to be dissolved and they're going to be washed away, right? I don't want that. So what I'm going to do is instead of a Bunsen burner, what I have is the following. I've got a little, lighter here, right? And the idea is to apply some heat, and usually you pull it through the gas flame 2 times so that you're able to hold it on your palm, and it's got to be hot, but it should not burn you. So the kind of the rule of thumb is it should be hot enough that you're just able to hold it without hurting you or without burning you. So that's kind of this was always the measure, and So I'm going to do the following now. I do not want to have it directly in the camera. Yeah. Okay. 1, 2, 3. I test it, and it's pretty hot. Okay. I'm still able to hold it, right? And this basically means that now the cells should be baked to the glass slide. Okay, still hot. and yeah, allow it to normally cool. And you are only allowed to do that once you're not allowed to do that when it's still wet. Okay, otherwise it's going to start boiling, and then the cells are gonna boil apart, right? But when it's dry and when you do that, then the cells should still remain intact. And what I'm going to do then next is I'm going to apply a little bit of methylene blue solution. Okay, on here. I allow it to, to stain this a little bit. Yeah, not everything is covered with methylene blue. You see that there's a little bit of yeah. And then I'm going to rinse it with some water. Okay. So this is basically the next thing that I'm going to do is I'm going to take this here, and I hope that this was actually already enough. And yeah, and I do it like you don't see it. And I take my washing bottle and just apply it on top. Okay, and not directly on the cells, right? Does you see the spot here? It's already stained, right? Yeah. To remove any excess. Okay, and I'm going to now carefully, dry the rest. I need some tissue paper. Yeah, you can actually see how it already stained it properly. Normally you should air dry it as well. Okay. Normally I have to air dry it, or I can use my coffee warmer, which I actually did not take out yet, which is a pity. So what I'm gonna do is I'm going to do that what you normally don't do. I'm flipping it around. Okay. You see some of the cells, yeah. So maybe I just have to wait for a minute or so until it completely dries, because as soon as if there's water present, you didn't see that. If there's water present, of course you do not want to mix it with oil. Okay, well, but it's kind of drying already. Maybe you want to shake it a little bit here. I should have actually prepared my coffee warmer. Okay. And then I'm going to explain you now, what I'm going to do now is once it's heat fixed, what you can do is you can actually directly apply immersion oil on the heat fixed cells. Okay, cover glass is not necessary. So this is it's starting to dry now. This is essentially the way that we've most of the time in the lab observed cells that were stained. Is no cover glass necessary, because the cells essentially are baked to the slide. There's still, you see, it's a little bit of moisture is still here present. Maybe I'm just going to try it anyway. And this should actually also work. Yeah. So this is actually something when I learned that the first time. This was also kind of new to me. What, you can actually use this without cover glass? Yeah, this was actually the standard procedure that we used. Not necessary. And I'm just checking again. You see there's still a small speck. It doesn't matter. It's just for demonstration purposes. I need to find my bottle of immersion oil again here. I apply it directly on here. I'm going to switch again to the stage view here. I'm going to lower this as before. I'm going to put the oil immersion objective in place. This one goes directly in here. It's got to be centered. I move it into the oil. Okay, and the scope view is here. Let's turn it up, and this is how it looks like. Okay. So these are the stained yeast cells. Okay. In oil immersion, heat fixed and stained. And I don't know, maybe we can actually move around a little bit. Maybe I'm able to find the place where the cells are not stained somewhere. I don't even know where this is. Yeah. So most of them are actually stained, as you can see. Yeah. And another thing that you might that you're noticing, I just want like to mention this. Look, there is now the cells are surrounded by oil. I'm moving around the slide, and the cells, they don't move, right? Because they're baked to the glass slide. Yeah. So that is, and this is the reason. So if you really want to, for example, to really want to observe some of the cells, quite well, and if you do not want them to float around vertically, and then essentially heat fixing is a technique that you can use. Yeah. Yeah. I'm just reading. Is heat fixing also possible with a coffee warmer? No, it's not hot enough. So this is. You can try it, but generally it's got to be much hotter. Yeah. Yeah. So this is simply to show you a little bit that essentially you do not need a cover glass for oil immersion microscopy. Yeah. You see over here, some of the cells here seem to be a little bit brighter. It probably also has to do a little bit with how long the stain could actually do its work. Now I'm going to go all the way to the side. Well, it's quite a lot. This is pretty huge. Maybe I'm trying to find the non-stained parts. Can't find it here. Here the cells are yet a little bit darker. Maybe here, because it didn't. The stain did not cover everything properly. Yep, maybe here. See, here it's a little bit brighter. Yep, here these are the non-stained cells, because the stain did not reach all of the parts. Yeah. So you see that staining actually, can improve contrast quite a bit. Yeah. Yeah. So this is a bright field. Again, I did not. If you want to see the whole thing in DIC, which is actually, I think, not really meaningful, because it's not blue on blue. Yeah. But this is how it looks, I think, much better. Yeah. So there's a very brief question. Can I now try this with a cover glass? How am I going to do that? What I could do is the following. I mean, that's just why not? I'm gonna try this. I'm going to now. There is no oil on here. Okay, I'm going to stay with oil. I'm going to lower this because somebody was just asking, and I have to do this now. And what I'm gonna do now is on here where there's the oil on here. I'm going to now drop a cover glass over it. And theoretically, it should not make a huge difference because the refractive index of the cover glass and of the oil is pretty much the same. So the objective should not actually see a difference, right? So I'm going to put it now like this on here. Okay. Now, because there is no oil on the cover glass, okay, I can now use the other non-oil objectives still, okay? There is no oil on the cover glass. It's only on the side here, so I got to be careful that I'm not touching the sides here. But just for the sake of comparison, I can now use, let's say, I don't know, 40 times or whatever. Yeah, there is now no oil. Yeah, I have to raise it again. I have to see if I'm able to find this, because it's kind of difficult. Here we go. Okay, so that is now the 40 times. So the cells, the yeast cells are now surrounded by oil because I simply dropped the cover glass on top, but I'm not using oil immersion. Okay, so let's do the following. Let's now add some immersion oil as well. Okay, just like before. I'm going to lower the whole thing. I'm going to put the oil immersion objective in place. I will now add a drop of immersion oil. Okay, that's kind of, and I'm going to raise it up again. And this is how it looks like. Need to make it a little bit brighter. And this is how it looks like, and there should not be a huge difference. Okay. So there should not be a huge difference because essentially it's still surrounded by oil, then there is a layer of glass, the cover glass, and then there's oil on top. So the light essentially should not be influenced a lot, okay? As it goes from oil into glass, back into oil. So the cover glass really does not serve a big purpose here. If you do not want to do heat fixing, what we can do, and I forgot which one it was, I think it was this one over here. If you don't want to do heat fixing, then what we can also do as a little trial before last trial before I start answering some of your questions, okay, why not try the following? I'm going to now add some of, I think these were the yeast cells, if I remember correctly. Okay. Yeast cells, yeast suspension. I'm going to add directly now the methylene blue stain. Very important is proper mixing. Otherwise, it's not going to stain, so I simply do it like this. Okay, you see I'm not using most of it. Cover glass goes on top again. Okay, cover glass goes on top. And let's now have a look again under the microscope. Stage view. Okay, as before, we remove this. I'm going to put this in here. I don't know if there's still enough oil on the objective or not. Otherwise, I'm just gonna add another drop here. And it goes all the way up again. Okay, and let's have a look here. And you see that is now, with methylene blue, and you see that the background does not look very blue. Okay, because it's not very concentrated, and the cells, they started to accumulate this. But I think you already notice something, and that is that some of the cells are out of focus. And why is that? Well, obviously because there are in liquid, right? And this means they're not baked to the glass surface, to the slide, and that means they're still able to move. And if I'm now focusing a little bit, look, I'm focusing. You can actually see some of the cells wiggle back, and the ones over here. I'm focusing, and you see this kind of makes them wiggle back and forth a little bit, again because of this slight pressure that the oil exerts on the cover glass. Yeah, but it actually also works. Yeah, but maybe this kind of illustrates a little bit this comparison of why you actually do heat fixing as well. Yeah, so or over here, for example, those cells here. Yeah, you see that they're still clustered together, and because they're surrounded by liquid, by water, that means that they're still able to move a little bit. Yeah, so this is simply something I just wanted to demonstrate to you. Okay, so I'm just also calling out if you have some remaining questions that you would like to ask. As long as they're microscopy related, I'll try to answer them. Please do start posting them, because in a few minutes, I think I will start to answer some of your questions. Yeah, I mean, I can actually comment on some of them already. Why does heat fixing not destroy the cells? Well, it destroys them in the way that they are killed off. Yeah, but essentially because the heat is not over a very long period. For this reason, the compounds in the cells do not have the time yet to burn. Okay, so this is a little bit like if you put an egg into a frying pan, for example, then it's going to be fried, but it still looks fine. But if you actually leave it too long or make it too hot, it's actually the molecules are actually going to start to break down, and this is when it starts to burn, and that's something that you should not do. But heat fixing will actually kill the cells because of the heat. Yeah, at least those cells that have survived the drying process, because not all cells survive the drying process as well. Yeah. Okay, so yeah, I see that you're posting questions. Thank you very much. I will basically be waiting a few more minutes. Yeah, and if you also have recommendations or of what you want me to do in future live streams, please post this as well. Okay. Yeah, see again, here you see actually the whole thing wiggle around. But again, I'm a little bit surprised still why there were so many. Or maybe I took the wrong. Could it be that I took the wrong petri dish? Maybe. No, this was actually this one. I'll try this again. It fascinates me. Those algae from the tablet, yeah, that they actually still contained all of these other things here. So I'm going to try this again. It's kind of fascinating. So let's take some of those again here. I'm just going to make a simple wet mount without. And let's just drop it in here again. Okay, at a low magnification. I hear that the questions are slowly coming. Let me try this again. Let's see. Thank you. Wow, there are lots of questions today. Here they are again. Okay, so these were basically the chlorella algae. There's lots of dirt and other stuff in here as well. This is actually not a good one. Yeah, I think the sample was indeed contaminated. Okay, look what I got here. Again, over a page. Of questions. And what I'm going to do now is I'm going to try to answer them. Okay. The very important. I just got the message, but I'll do this now. Thank you very much. So what I'm going to do is the following. There's been a request to quickly show the cleaning process. Okay. So this is the 60 times, which I can just remove. So what I do for cleaning the objective is I use some. You see the philosophy goes apart a little bit here. Some people are saying you should not be cleaning the objective because you'll be using it a lot anyway every day. I mean, and if, for example, if you're in a lab where you're doing a lot of the routine analysis, they say there's no point in cleaning it. You just basically leave it on there, especially if you're using synthetic immersion oil. Then essentially there is no danger of it solidifying. But I always clean it off. And what you do is you use the lens cleaning tissue paper. Here it is. Yeah, which looks like very, very thin. And then what you do is you just what I usually do is I just very slightly remove any excess oil because the oil will collect dust. Okay, and if there are any dust grains in there, then and you clean it, then it actually might scratch the lens a little bit. So, but that is essentially very lightly. I talked to the company that sold me the microscope, and he said is you do not have to be afraid of cleaning the lens. And he actually told me that because the front lens actually goes inwards, it's concave. He said sometimes what you have to do is what they actually recommended is take. I show it over here. Yeah, he said take a. I just showed over here as a demonstration. The front lens actually is concave. It goes inwards. And he said if you just wipe over it, you're not able to properly clean the lens. So he actually told me you should take a little bit of this lens tissue paper, wrap it around your fingernail, and don't be afraid to actually go in and carefully clean it out as well. He told me that those lenses actually are, how do you say, more stable, more stronger, and they're able to withstand more than what you might think. Yeah. So there's many people are actually the lenses can take more aggressive cleaning than what sometimes people are willing to do. Yeah? So that's kind of what I was told. Yeah? So he said, don't be afraid. Don't use any hard objects, obviously, right? But if you do something like this, apparently it's not a problem. Yeah, don't be afraid to kind of dig in and scrape out the dirt, so to say. Yeah? I was told. I still didn't do that because I was too afraid. Yeah, but this is basically how it looks like. Yeah, and again, stage view, and and basically, if I don't see anything, then I say it's fine. Okay, so that's basically what I'm doing. And now I'm going to go back here. I touched the 40 times. Okay, yeah. Luckily, because of the capillary action, it's how do you say, it's kind of absorbs it a little bit here. Okay, so I'll put this away, if there is a little bit of oil on the other lenses, then you can use some alcohol to carefully clean it off, but it should not be actually dipped in oil so that there is kind of too much oil which can actually go into the cracks of the lens. Yeah. So desk view, here we go. Okay, I did not read those questions yet, so I don't know if I'm able to answer all of them. Okay, I'm just saying, but I'll try to comment on them. This is what it was at the beginning. It looks, are they immersed in oil? This was, I actually also answered this. Yes, they are. About an old Zeiss microscope from the 60s, there was an additional bottle of cedar oil. Can this be used for immersion? Actually, as a matter of fact, that this cedar oil is actually this, many of the natural oils are actually cedar oil. And if you smell them, then they will actually smell a little bit like the cedar wood oil. Yeah. So the natural, without knowing much more, that's what actually many natural oils are made of, is cedar oil. Okay. I got an Olympus 40 times 1.3 NA oil objective, a 40 times oil objective. I absolutely love it. Amazing clarity as an upgrade to not so expensive microscope. Have you tried oil below 100 times? No, I have not. Short background information. When I bought the microscope here, I just show you again the scope view, the stage view here. I've got here a 60 objective, and when I contacted the company, said I want to have a 60 times objective, he actually gave me two options. He says, do you want to have a dry objective 60 times, or do you want to have an oil immersion 60 times objective? And I basically said I want to have a dry objective, and the reason why is because I personally consider the non-oil objectives more versatile. I simply use them more often because I don't have to use immersion oil. Yeah. So for quick observation and for making those YouTube videos and for showing things, simply the non-oil objectives are simply more practical. Yeah. And that's the reason why I have not tried it. That's the only reason. Yeah. But it's a possibility. Huh? Do you ever think about making a laboratory team with all of your subscribers, and then you could sell real laboratory products to all of us like UPRL and so on? This is actually something that a couple of years ago, I was actually thinking about because I had problems actually getting microscopy equipment. So I thought, well, actually it would be kind of cool to have a separate kind of microscope laboratory shop where you can buy all of the things from one shop. But this basically would have taken a lot of time, and it would have distracted me actually from making microscopy YouTube videos. So what I've done basically instead is I collected some how do you say, products over the Amazon affiliate, and that's why I collected them and made an Amazon affiliate shop. And there are, however, also basically, I'm not affiliated with them, but if you're living in Europe, Brunel Microscopes from the UK has pretty much everything that you want to have. Yeah, and they're pretty good stuff as well. Yeah, so it's Brunel microscopes. I'm not affiliated with them, but I ordered already a couple of times from them. And if you have any questions, then they're also able to actually, answer them. Yeah, maybe you could make a video on how to clean slides and cover slides. I throw them because I'm immunosuppressed and I'm afraid to cut myself. That is indeed a point. Okay, and I will show you the following. Those slides that I have here, I cleaned them yesterday. And indeed, there is, of course, the danger of cutting yourself. And this can happen, especially with the cover glasses. So what I have done is the following. And because I want to reuse the slides as well, not only for environmental reasons, but yeah, they're not so expensive, but I simply want to, and sometimes I don't put any problematic materials on here, just some plant cross sections that are easy to clean off. And so for this reason, what I have done is I bought slightly better slides, and those slides, they have basically polished corners, and maybe you're able to see this over here, that over here also the edges are cut at 45 degrees. Okay, so this basically means that they are much, better to handle them. And if I kind of clean them, then there is actually with those not so much of a danger of actually cutting yourself. The issue is, of course, always different with the cover glasses because they can break. They do break. Yeah. So sometimes what I do is actually I will throw them away, the cover glasses away, and I will simply only rinse those slides. And another thing that I can recommend is, this also kind of works, is you just take the slides as they are, you remove the cover glasses and you put them into your dishwasher. Okay, and then most of them come out pretty clean, some of them not, you just throw those away. But actually with dishwasher it works quite well. I personally would say that if you're using, I don't know, just plant cross sections or non-dangerous, non-infective material, it should not be an issue. But if you're immunosuppressed and if you're observing water samples or whatever, it's just be a little more careful. So I can understand this. Yeah. In order for me to reduce my danger of injury, I decided to buy slightly better slides. Okay, and they're polished here, and they have a 45-degree angle over here, so they're not quite as sharp. Yeah. Maybe a video on how to clean slide. Okay, how long will the heat-fixed yeast cells stay preserved? Can I keep them for several years? No, only if you add. You can only if you add a mounting medium. Okay, so this is the thing. The heat-fixed cells themselves. I've got some other slides over here. Yeah, that I prepared. It's not yet heat fixed, but I mean, if you heat fix them, they can be scratched off again. Maybe not as easily as the non-heat fixed ones, but this is not a permanent slide. So if you want to do that, you can dry it. You add a mounting medium on top, and you add a cover glass, and then it should be preserved for years. Yeah? But just heat fixed alone is not stable enough. Physically not stable, because if you scratch over it, you still can basically scratch the cells off. Yeah. So that's the thing here. Okay. But aren't there objectives that are made for immersion oil without cover glass? Wouldn't that mean that the cover glass correction in general doesn't matter? That is correct. So basically, I don't know if I have one here. Some of those, look, I explained this using this here. Okay, here, maybe you can see this because it's a nice example. This is not focused. Let me go down here. So the numbers over here. This exactly answers your point. So achromatic 100 times magnification, numerical aperture 1.30, oil immersion. Look at this, 160 millimeter tube length. That's the standard. And then there is a dash here. Instead of a cover glass thickness, they said it's irrelevant. Normally there is a 0.17 written over here, which is the cover glass thickness. But for oil immersion objectives, the cover glass thickness is irrelevant because the light does not see a cover glass. Because it goes from even if you use a cover glass, there is oil on top of it. Yeah, so the thickness of the cover glass is irrelevant. Yeah, and that's why some oil immersion objectives actually, instead of having a cover glass thickness, they simply have a dash here, just like over here. Yeah? And just to show you as in comparison, I have another one over here. Yeah, it's a 10 times. You see over here, it's a 0.17. It's written on here. Your cover glass is important. Yeah? So this is basically indeed the case. Cover glass correction doesn't matter. Yeah, it's correct because it's in oil anyway. Yeah. Is heat fixing also possible with the coffee warmer? This is something that I think also already answered. It's not hot enough. My coffee warmer goes to 80 degrees. I think it's got to be hotter than that. Yeah, and one might try, but yeah, generally I think with the flame it's a little bit hotter. How about using a simple lighter to heat fix the cells? That's possible. This is actually a lighter. Okay. I think you just have to be careful that there is no soot. So candles, for example, are not so good. Try this as well because sometimes there's soot depositing beneath it, and that's something you don't want. Yeah. Can you also heat fix algae and other water microorganisms? Sure. I will just tell you the following: that the problem with other water microorganisms is that usually they will start to lose their shape during the drying process already. So, for example, if you've got I don't know some delicate water microorganisms, and when they dry, they actually might lose their shape. So this is something that you might take into consideration as well. Certain algae, if I would say cyanobacteria, okay, they're not really algae, but okay, it might work. But generally, very often algae also a little bit thicker than some other cells, so you have to be a little bit careful of whether actually they make contact with the glass slide. I would say that's probably the criterion here. Could you try again and use a cover glass? I have done this. Okay. Can we heat fix plant cells? You think no? This is generally not done. Okay. The point of heat fixing is that essentially individual cells are baked to this glass slide so that you can stain them. I've never heard of heat fixing plant cells. For heat fixing plant cells, you use usually some chemicals, other chemicals that and then you dehydrate them and then you embed them. But generally, heat fixing is mostly for single-celled microorganisms, yeast, and especially bacteria. Yeah. What is the name of the gas tool that you use to heat fixing? Honestly, I don't know. This is just some kind of a fancy lighter. Yeah, I think it's even refillable. Yeah, and the nice thing about this is I have to be careful that I don't burn anything. I don't know if you're able to hear the sound, but you're not able to hear the flame. The pressure is a little bit higher, yeah. This basically is like a mini Bunsen burner. Yeah. It's the same question with a simple lighter. I have also done that. It should work. Yeah. By the way, I've even heat fixed once using the kitchen oven, the hot plate in the kitchen. I carefully placed it on there, and this actually also worked. Yeah. Okay. Yeah, it's too funny. I cannot resist looking at the live comments. That's correct. Thank you. I've been looking at the objectives from size, and some are made with and some are without cover glass correction. That's why I was wondering. Yep. Mm-hmm. Yeah. So, why does heat fixing not destroy the cells? As I mentioned, it will kill the cells, right? But it should not destroy the structure of the cells too much. Yeah, if you exert too much heat, you're going to burn them. It's going to start actually turning black and they're broken. Yeah. Do you have any tips on getting thin gill sections of mushrooms? The gill sections of mushrooms. I know what you mean. You'd have to microtome them, but how are you going to microtome them? What I have tried once is you have tried to actually take some tweezers, and because the gills of the mushrooms which form the spores are relatively soft, I've actually was able to compress them as well. Yeah, so that means I've taken some of the gills, small samples of the gills of a mushroom, and I tried to compress them between a cover glass and a microscope slide. And then sometimes you're also able to see the parts that are actually making the spores. Yeah? Otherwise, you'd probably have to embed it somehow and then microtome it. Okay, but that is quite a fancy and complex procedure, I would say. Yeah. Sometimes what people do is to take a mushroom, they put the mushroom on a piece of paper, and then they use an object and they knock against the mushroom, and then this kind of where you have the gills is where the spores then start to deposit, and then you can actually have a spore pattern, and you can put that also under the microscope. Yeah. Continuing from the nematodes last week, I know that annelid worms are segmented, whereas flatworms and nematodes are not. But how do flatworms and nematodes differ from each other? Let's put it this way: these are different phyla, and I would say probably flatworms. Let's put it this way: there's several differences. Flatworms, what they have is, for example, planaria. They have a one mouth. It's the feeding tube that they have. It's not really a mouth. A feeding tube, and they basically will suck up the food using this feeding tube. It will be digested, and then they're kind of spitting out the non-digested materials. And nematode worms, what they actually have is they have they seem to have a digestive system running through it as well. Yeah, so there are different phyla, and both of them we call them worms. But this does not mean just because we call them worms, both of them, that they're actually necessarily related to each other. So that's a little bit the thing here is that maybe the term worm implies a greater similarity than what actually is there. Yeah. So there are a whole range. But you know what? If you want me to, I can actually, why not do a little segment, a little live stream on different types of worms, like for example, Iolosoma, which is a segmented worm, and then the nematode worms. We've got some vinegar eels here as well, which are basically non-segmented worms. But essentially, they're two different phyla, in the animal kingdom. Yeah. So people. It's already, again, almost 1.5 hours. Maybe there are a couple of questions that arrived last second. Let me quickly go back here. Yep, I think most of the question... there's a comment here. I'm reading 19th century publications like Zeitschrift für wissenschaftliche Zoologie, and I wonder... So that's a basic Zeitschrift. It's a magazine for scientific zoology. In German, and I wonder if I could make observations like they did. Any recommendations in that regard? Let's put it this way. We got all of the equipment that we need, but if you look and read some of the original publications from even the 19th century, I was sometimes myself, I'm quite surprised how advanced they were in some of the observations. I'm wondering myself, how in the world did they figure that out? And very often, and this is, how did they actually discover all these things? And then when you do a little bit more research and you discover that this was not, many of the discoveries were not just simple observations under the microscope that they've done in one evening, but sometimes these were quite extensive studies until they've uncovered certain things. So we've got the equipment these days, but I think what we might not have is maybe the whole time and a whole team of people actually doing the observations as well. All of the stainings that have been done, and then you repeat the experiment and over and over and over again. So it must have been some of the observations that we're just reading these days must have been quite tedious at that time. Yeah, so I would say we might have the equipment, but the know-how is also there, and the universities they're doing that. But as hobbyists, it's a question of whether. We are willing to invest all of this time to repeat the experiments. And some of the chemicals that they used were not that, were problematic as well. Yeah. Maybe you should also show the cleaning process of the... I've done this now. Would it be impossible to have algae without any contaminants? Well, let's put it this way. If you are doing research at a laboratory, at university, and I've been doing research using in bacteriology. You have to work with pure cultures. If there are contaminants in there, it's a no-go, because if you do chemical analyses, then contaminants might, well, it certainly will give you wrong results. For example, if I want to determine the chemical composition of a bacterial culture to determine which bacteria are they, and I've got a contaminants in there, I'm going to get completely messed up results, right? So having pure cultures for standardized experiments is extremely important. Yeah. Right now, what I'm doing right now is I'm not doing those experiments. I'm just basically I'm not even working sterile, as I've got my glass jars with water samples and whatever not in there. And I'm happy if they're not pure cultures, if I've got a lot of interesting things growing in there. So my objective is entirely different than essentially the objective that you have at a university, where you need to do systematic reproducible experiments. Yeah. Can you put yogurt under the microscope? Yes, I have done this as well. You see yogurt bacteria. That's yeah. As a matter of fact, especially homemade yogurt looks quite. But why not try this next time as well? Yeah. Okay. I watched touched the 40 times. Okay. The problem being in the UK is you have to pay extra taxes. Oh, I see. Okay. Yeah, it's probably value-added tax. Yeah. And I bought a cheap electric ceramic hot plate that the chefs use in the kitchen and use it at lowest power setting to dry permanent slice. Yeah, that's also cool. Okay, thanks for picking up the topic. Okay, yeast prep shows the real advantages of oil immersion. You could clearly make out the internal cell structures. Yes. Okay. Have you ever observed wild yeast used for making sourdough bread? No, have not. I know that there are sourdough bread preparations, so maybe it's possible to see them in there. But usually, if you want to observe, let's say, wild yeasts, wild bacteria growing naturally somewhere, usually it's a little bit difficult because if they are in a significantly lower concentration than the object that they're growing on. I'll give you an example. For example, if you want to make wine, in the past, what they've done is they simply took the grapes, they made grape juice out of it, and they simply let it ferment without adding any yeast to it, because the natural yeasts that were present on the grape caused the fermentation. Okay, so no problem. But the problem is that if I now make grape juice and I've put it under the microscope, I'm going to see way more grape cells than I'm going to see bacteria. No, than I'm going to see the yeasts. Okay, and so this identifying those yeasts from a natural environment, that is a difficult thing, because they're often at a lower concentration than the other cells that you've got there. Yeah? Maybe last question, is it possible to use slides on inverted microscopes? As a matter of fact, why not? I guess it depends a little bit on the stage of the inverted microscopes. Inverted microscopes are used so that you can take a whole Petri dish. I have a small one over here. Yeah, and for example, when you grow cells in a Petri dish or in cell culture, what you do is you basically place it on the stage in an inverted microscope. The objective, okay, is on the bottom. Okay, so this allows you to observe the specimen without actually having to make a slide. You just leave it in the Petri dish, you put it on top, and you observe it. Oh, of course, I get it. You see the thickness of the glass or the plastic Petri dish and so on, and the working distance. So you're using different objectives with a larger working distance. But essentially, I see now, I've not tried this, but I see no problems now if you're, instead of using a Petri dish, why not use a slide? But then if you're using a slide, then you'll probably also be observing the specimen through the glass slide itself. Okay? Yeah. So I see no reason why it shouldn't work. Yeah? But maybe some of you know a little bit more about this. Yeah. But you cannot have pure algae, right? Why not? If you essentially grow them in a pure culture, you can. And if you isolate them, and it's the same thing that I've done in when I did my studies at the university, The first task is when you isolate cells from the natural environment, you have to get them into pure culture. And that takes some time. Yeah. So, and then there's always a danger that they become contaminated. Yeah. So this is actually something that you have to do. You have to maybe take individual cells and you have to try to grow them in a condition that they multiply, and then you have hopefully a pure culture. Yeah. So, yeah, but more often than not, the thickness of using a petri dish makes it difficult to observe things more properly as compared to a slide. Yes. Yeah. I probably would agree. However, one of the things that I also want to mention is that if you're using inverted microscopes, for example, in so-called in a cell culture, where you're growing, for example, human cells, then very often these cells will not swim in the medium, but they will also stick to the inside bottom surface of the petri dish. And this basically, again, makes them basically flat in one plane, and this makes it, again, easier to be observed. But I guess it depends very much on what you want to observe. Yeah. So this is but I can imagine that maybe the thickness of the petri dish and the distance from the objective might be actually an issue. Okay, people. What I would like to do now is after 1.5 hours, I would like to thank all of you. Especially, I would like to thank Michael for the contribution. Michel, I hope I pronounce your names correctly. Okay. Tubekin, hope, and Trick Track and Pat Burnett. Thank you very much. I hope I really did not overlook anyone. I want to thank you very much for your contributions. I will use these, of course, to buy microscopy slides, and then I can show you again a few things. Otherwise, I would like to simply say bye-bye. I hope it was interesting. Hope that we all learned from this live stream, including myself. Every live stream is a little bit different, even though I've sometimes been doing the things already before. It's always a little bit, there are always a few new things and surprises that appear. Thank you very much for your comments, for your questions as well, which basically makes also the live stream interesting. I'm going to think again of a topic for next week. I think I'm just going to say, just watch also my other videos that I make. I try to make at least 1 or 2 videos additionally in this live stream. Besides the live stream, I try to make at least 1 or 2 additional videos per week. If you have any recommendations or suggestions, then feel free to voice them. Otherwise, I say all the best. Happy microbe hunting as always, and see you again next week. Okay, bye-bye. Bye-bye.